hello everyone, very nice to meet you today. So thank you, Chayong, for giving us this opportunity to share the capability of Eurofins Discovery. And today our topic will focus on the safety pharmacology portfolio of Eurofins Discovery services. So I'm Chun-Yao Lee from Taiwan. So now I'm the lab director of molecular pharmacology department in Taipei site. So I will show you the safety pharmacology portfolio for the whole Eurofins business unit. Okay, so before the meeting, I just like to give you a short idea like how Eurofins works and also for each site's capabilities. So I'm from Taipei site, as you know. So in Taipei site, our brain span labs. So we provide different kind of in vitro off-targets, therapeutic-based portfolio, and like many binding enzymatic cell-based and SPR-based binding assays. And also we have in vivo capability like a DMPK or some disease animal models. And also in the US, we have a different site from San Charles, Fremont, and San Diego. They provide different kind of cell-based or phenotypic assays and also some GPCR services. And in France, UK, and Spain, our European sites, they also provide some profiling and some protein products and even so some medicinal chemistry and chemical synthesis capabilities. So in Eurofins, we are a big family and each site we support each other and we have covered all the territory of different capabilities of different therapeutic areas. So that could provide you the overall whole service as a package. Okay, so for today's presentation, this is the outline. So I will introduce you how we provide the safety pharmacological services and how this kind of service can help you for your drug discovery. So I'll give some overview on safety pharmacology. So this is a science on detecting adverse effect liability or predict potential risk and counting safety margin for medicine. So this field is very important during drug discovery, especially for the early phase. And later I will introduce you some case study by our safety screen panel. So how clients use our panel to evaluate their drug and adverse effect and risks. And later we'll go through different fields like CNS safety, so including drug abuse and seizure liability, and cardiovascular safety, so-called CPAR related to arrhythmia, and then cardiogenesis safety. And in the end, also we'll show some in vivo capabilities in our site. So let's start from the safety pharmacology overview. So I think many people already know the definition of safety pharmacology, or we call secondary pharmacology profiling. So when we have a compound, a drug, or a prodrug, or even its metabolites, we are hoping this drug to have a hit our intensive targets, like we decide the drug to hit the GPCR or hit a certain enzyme to have their pharmacological effects. So this is our purpose. But during the discovery, sometimes we find the drug is not only hitting our primary target, but they have some off-targets hitting, or we call this a secondary effect, secondary hitting. So that will cause some problems or raise some issues to us because this is unintentional hitting. So we have to think about if this secondary effect is beneficial, neutral, or deleterious. For safety pharmacology field, mostly we are worried about, well, we have some concern about the safety or further risk, potential risk. So we are thinking maybe the on-purpose targets will be harmful. But sometimes it could be beneficial to reveal a new window or vision to our compound. So it depends. But then most of the on-purpose hits could produce some concern. So we need to evaluate what is the off-target and how we can address on this issue and how we can further modify our chemical structure to avoid the secondary effects. Because this kind of secondary effect, if this is harmful, maybe they can produce some potential risk like adverse effects or get some harmful to the cell or to the whole body, and maybe will induce a termination of the whole project. So in the early phase, we really need to think about the secondary effect to define if it's a harmful or it's neutral. And later on to design our drug accordingly. So from the guidance we know, the FDA guidance we know, the safety pharmacology study, the main concern will be about our core battery system. So what is the core battery system? So-called is our central nervous system, cardiovascular system, gastrointestinal system, renal system, respiratory system. These systems are very important to our vital signs and our normal physiological functions. So if we have some secondary effect which will affect our core battery system, that will probably produce some high-risk harmful effect, or we really need to assess the risk factor in the future. So if any compound they have the secondary effect and related to the core system, that will be very cautious and we really need to pay attention. So in the right side, you can see, in the right side, you can see according to the instance and the impacts, we have 4 different areas to the risk level. So, and also from different colors, we can see different fields like cardiovascular, neuron, respiratory, renal, and GI. So we have some secondary effect or adverse effect. They have low instance and low impact. So that would be less, maybe just cause some minor effects. Or they have a high incidence but low impact. So something like if we take some medicine, we have some comfortable in our GI function, or we feel like nausea, vomiting, or we have change of our heart rate and blood pressure, but they are not lethal. But for some adverse effects, so we can see here they have a high impacts, like they have some retinal dysfunction, they have a neuropathy, or they are leads to the seizure or drug abuse liability, and that will be cause some high impacts. And even more, we have a high incidence and high impact like a QT prolong and pro-arrhythmic effects. That will really cause some high risk for this drug discovery. So this kind of effect we should really take into consideration and evaluate this very seriously. So according to the guidance, this is the journey of the drug discovery. So we have a safety guidance, ICH 7A, and we have a different category, like we have our primary therapeutic targets, our primary effects. Like this is our primary hit. We like to have the therapeutic effects. So we'll do some biochemical, functional, or even phenotypic assays. And later on, to finalize our study to understand the mechanism and how it shows its effect in the different therapeutic areas. But meanwhile, we'll have some secondary pharmacology, or we say the off-target profiling. So we'll use a lot of like biochemical or functional tests that we screen a lot of different molecular targets, and to find, for example, some researchers, they will do some safety screening, hERG channel binding, or evaluate the drug abuse potential to understand how the secondary effects or the off targets will make some further effect during the discovery. And after that, we have some safety pharmacology, in vivo safety pharmacology. So we use animal models really to evaluate the safety issues or concerns on the real animals. So either we can do the safety or toxicology in non-GLP or GFP required studies, and to finalize the whole journey of safety pharmacology study. And during this process, in vitro system plays a very important part because it's fast and cost-effective and can really cover and include a lot of different molecular targets by biochemistry or functional assays. So even cover like a lot of tissues, cells, organelles, and even on the molecular level, like receptors, ion channels, transporters, and enzymes. So in vivo study plays a very important part during this journey and help us to identify the targets. So here I want to just show you some industrial practice of safety pharmacology. So in the right one, you can see the very famous paper published in 2012 in Nature Drug Discovery. So this paper is contributed by 4 very famous pharma from AstraZeneca, Novartis, GSK, and Pfizer. So their scientists and their research team defined 44 very potential targets they probably can produce a high risk in after adverse effects during drug discovery. So they figure out these 44 targets will be important, and at a very early stage, researchers should pay more attention on these targets because they are related to the core battery system and may produce some higher risk and adverse effects. So in our safety panel, we include these 44 targets as our safety panel. And later I can show you how we use this panel to do our case study and how this will help you to identify your off-target hits and secondary pharmacology. So in this paper, it's also mentioned about some early terminate projects and drug candidates from different periods, like from 2004 to 2010. And also they include some clinical candidates discontinued and some withdrawal drugs. So according to this profile, people can find that if your compounds with higher promiscuous rate, meaning a high target hit rate, meaning like you have more secondary targets, and that will be more possible your compound or your project will be discontinued, or even the compound already pushed to the market, there will be some risk of a withdrawal. So you can see the clinical candidate discontinue and withdraw drugs, they have a really high hit rates of secondary targets. So later I will explain you how we approach this by our case study. And another research from 2018 saying about usually the pharmas, how they leverage the resources from CRO and how CRO plays an important role in industrial practice of in vitro off-targets. So they made a survey on 21 scientific experts from different pharmas or industries and how they have their data and how can they approach their studies. So you can see most of the pharmas and experts, they use both way, like hybrid approach. They have internal capabilities, but also they use CRO to help them to do the off-target profiling. And mainly on a lot of small molecules and also probably some large molecules as well. So it's very rare a pharma or a biotech, they use their own internal capabilities to approach the off-target profiling. Usually they can leverage the resource from CROs because in CRO we have built up a lot of different molecular targets. We can cover a very wide range of targets and provide different kind of from biochemical to functional, even phenotypic assays. And from internal resources, sometimes it's not enough to really have all the experiments or all assays by their own. So very often, pharmas, they will use CROs to complete at this early stage of target profiling for their drug candidates. Okay, so next one, I'm going to show you how we use our safety screen panel case study with some already marketed drug and how we can find the result and how we interpret the result with already known secondary side effects. Okay, as we know from in the discovery journey, we have different stages. So we have when we have a compound or we have a candidate, and we need to find a hit, and then we have go through the whole journey like hit to lead optimization, and candidate selection. And during this journey, we need to identify the potential hazards, and maybe later on during the structural modification or SAR, and we can eliminate our hazards, and later on do a lot of functional assays to understanding the mechanisms. So our safety panel will cover different territories and provide a lot of different choice to our clients. So firstly, we have a tier 1, 2, 3. So the 1, 2, 3, they cover different numbers of targets. So the tier 1, as we mentioned, the 44 main target proposed by pharmas. So we have the first stage, tier 1 safety panel, so safety 44. So they cover all suggested targets provided by the pharmas, And if clients, they have some concern, they want to step forward their first step to the safety pharmacology, they will start from the tier 1 safety 44. But sometimes our clients, they have more concerns, or they think about, I want to know more about if I have other off targets except from the 44. So we have a tier 2 safety 87. So they cover even more GPCR and enzymes to provide a better view if you have other off targets. And we have also tier 3, so it's like spectrum screen. So some clients, they are very strict and very careful about, they want to exclude all the possibilities on the off target. So they will do the spectrum screen panel. So we have more than 160 targets. So it's kind of really overall screening to understand and also to exclude other potential off target, even they are not very often. But to be careful, some clients, they want to see everything. So we can also provide a tier 3 spectrum screen panel. So coming back to the safety pharmacology. So in this case study, we pick up our typical safety screen and ADR enzyme screening to approach our case study. So here in our adverse reaction enzyme panel, so we have 30 known kinases, including cytochrome P, protease, PDE, NOS, and so on, and also 21 kinases like CAM kinase, MAP kinase, and tyrosine kinase families. And also we include our safety 44 and 87. So actually our 87, they cover all the target in 44. So some currently they just order 87. That will be enough to cover all the 44 target proposed by the big pharmas. So in the safety screening panel, so we have enzymes, GPCR, ion channels, transporters, and NHR. So overall, we screen like 139 total targets to figure out the compound if they have a off-target effect. So let's go to the next one. So in this case study, we pick up the compound loperamide, so-called imodin. So this is a very old drug approved in 1976. The indication is for anti-diarrhea. So I think many of us already we experience diarrhea, and sometimes doctor just give us the imodin to stop the diarrhea. So the primary target of the loperamide is on the opioid receptors, including mu, delta, and kappa receptors as an agonist. But in 2016, the FDA just announced an alert saying the loperamide may probably cause some heart problems. So during the market period, already 48 cases have been reported with serious heart problems associated with the use of loperamide. So by this case study, we would like to put this compound, loperamide, to our safety panel to see what we have and how we can interpret the data. So as mentioned, we include overall total 139 ADR-associated targets included by our adverse ADR enzyme panel and safety screen panels. So for each target, we can classify them into different areas like CNS, CV, endocrine, GI, immune, oncology, respiratory, and so on the others. So we can know each target is related to different areas, or some targets they are overlapped or they share by different systems. And we use this compound as 10 micromole as testing concentration. And if the inhibition rate over 50%, we define it's a hit. So under this definition, we start our safety screen panel and we see what is the readout. So this is the result, heat map of the result we can see here. So first of all, we can see loperamide indeed, they have a very high hit on the opioid receptors, including delta, kappa, and mu receptors. This is very consistent with our already known knowledge. But meanwhile, we've also find the other target is including alpha beta receptors, hERG channel, sodium channel, calcium channel, and dopamine receptors. So that's an issue. Why we have other off targets and how this off targets works. So later on, we just perform some functional assay by cell-based platform. So by our functional cell-based assay, we can identify if this hits because we have a binding data. So we know, okay, they will bind to these receptors. And after then, by cell-based assay, we can identify if they are agonist or antagonist, and also to measure the IC50 on the cell-based platform. So in conclusion, we just find loperamide is not only an agonist of opioid receptors, but also they are weak antagonists against alpha-1b, alpha-1d, D3, and calcium 1.2. So this is very useful information. So we already know loperamide works on opioid receptor, but now we all know the off targets will be from the other receptors. And when we compare to the different different areas of the related risk, so we can compare. So by this figure, you can see on targets, we have opioid receptors. So the indication is like acute and chronic diarrhea. But according to different receptors or ion channels, we find like this alpha receptor, dopamine receptor, calcium channel, beta receptor, serotonin receptor, and HERG and sodium channels. They are related to different cardiovascular adverse events like hypertension or arrhythmia, heart failure, cardiac arrest, or some cardiac stress, or even some thrombosis and arrhythmia, especially for HERG channel. This is very important. So according to this data, we already know that, okay, we have a good indication like an anti-arrhythmic effect of loperamide. But the other off-target could explain the phenomenon already we observed from the side effects. They have some heart problems announced by FDA. So by this example, we can explain, okay, so of course, because they have some off-target and all these molecular targets are related to cardiovascular field. So that's why they may cause some heart problems after if you use this medicine. So this is a very quick example for us to study, a case study to understand how our researchers, scientists, our clients, how they can use the safety screen panel to evaluate the risk of their compounds at very early stage. So this is the position and impact of secondary pharmacology. So according to the result, we suggest if we want to do the safety screening or the safety, the secondary profiling, we suggest our researchers to do it as early as possible. Because if we find this is at late stage, so that would be very costly. So if the early stage during the screening period already we find there are some potential, we'll know this and we can modify or adjust structure accordingly. And also we suggest to screen the target as wide as possible. So basically we have 44 very important targets published by the paper. But if we want to be very critical and very strict, we really suggest to screen as much as we can and to exclude all the potential off targets. And later on, we can suggest our client to do some dose response curve to identify IC50, because if later on, if you like to perform some functional assay or some mechanism studies, you need this information. And also by our functional assay follow-up, we can identify the compound if it's an agonist or antagonist on the certain receptors. So they can provide some functional evidence on how it works. And later, of course, our internal scientists or expert in the pharma, you should like really interpret the data by your own or by some suggestion from external experts to evaluate if you want to continue this project or how you can adjust to modify your structure to avoid this off target. But in this slide, I just like to mention is off target is not only harmful or make some problem. Sometimes this kind of off targets can reveal or open a new window for the projects. So for example, recently we have a lot of theory about multi-target drugs. So maybe before we think the drug is a dirty drug, they have a lot of off targets. But some research they mentioned about this kind of multi-target, probably they can contribute to the synergistic effect on the different targets and have better therapeutic effects. So this is also worth to pay attention to. And also sometimes even we find some off target, for example, if our principal project, we want to do some drug discovery and aiming on certain purpose, but maybe this project failed, but we find some off targets, and this off target can potentially provide another opportunity to revive this project. Maybe, for example, if we are working on the dopamine receptor treating Parkinson's disease, but in the end we find off-target in serotonin transporter. So that's finding could be contribute to another new opportunity, for example, for antidepressant drug discovery. So the more we know, especially on the off-targets, probably we can have more opportunities and more options for our projects. So just like to mention, not only for the harmful secondary effects, sometimes the off-target could be beneficial to our studies. Okay, so next one, I'm going to the CNS part. So I just like to show you what CNS service we can provide regarding the safety pharmacology. So we have a CNS target panel. Actually, we select all the most biological relevant CNS targets into this panel. So if you are studying in CNS, no matter in the therapeutic areas or you want to do CNS off targets, you can think about to start from the CNS target panel, because we have a 94 assays all included in very important CNS receptors and ion channels. So this will be helpful to your study on like neurodegenerative disease, psychiatric disorders, or emotional disorders, and also like cognitive problems, Parkinson's disease, Alzheimer's disease, and so on. And there are 94 biological relevant targets, including enzyme, GPCR, ion channel transporters, and nuclear hormone receptors included CNS panel. So if our client is focusing on CNS field, sometimes they can start from the CNS panel. And next one, I want to show you the drug abuse safety panel. So this is more specific for drug abuse safety and the safety liability, because a lot of compounds, they have a potential risk of drug abuse. So we already know in the brain circuits, like basal ganglia, nuclear basal components, amygdala, and prefrontal cortex, that relate to drug abuse. And the all drug abuse mechanisms, they relate to different kinds of classes, like opioids, alcohol, nicotine, CNS stimulant, cannabis, hallucinogens, inhalants, and BZT and barbiturate related. So they are related to different kinds of mechanisms on different levels and also on different brain circuits, and they can potentially cause some drug abuse potential. So during the drug development and discovery, we really need to pay attention if our compound have a risk of drug abuse. So why it is important? Because FDA already provide a guideline to assess potential drug abuse. So in the guideline, we mentioned about some specific neurotransmitter pathway like dopamine, serotonin, GABA, opioid, cannabinoids, MNDA, and some ion channels and transporters. They should be related or to included into drug potential assessment. So they suggest we use radioligand binding assays at starting concentration 10 micromolar to test this potential molecular target. If they have a hit, so that probably indicates a potential drug abuse risk. So further assessment and evaluation should be necessary. And this is the assay list in drug abuse safety panel. So we include all the related molecular targets for drug abuse, same including enzyme, GPCR, channel, and transporters. And last one for the CNS safety, we like to mention about the seizure liability. So for some compounds, actually they have some proconvulsive effect, especially for the infantile or juvenile. So when you use this compound, they may probably cause some seizure. And this is what we also like to avoid. So for the seizure implication, usually this is related to ion channel. So we list out some relevant ionic channel like calcium channel, GABA receptors, glutamate receptors, potassium channel, sodium channel, and so on, to evaluate if a drug is proconvulsive. And if we have some hit on this, probably that's where we have to think about if we have proconvulsive concerns. And later on, if we have some hits, later on we can also use our in vivo seizure models, like we have a maximum electroshock animal models, PTZ-induced seizure models, or insulin-induced seizure models to see if this kind of drug is really proconvulsive. And if clients really would like to understand more how the compounds works on these ionic channels, we can ship your compound to some trial site to perform more dedicated electrophysiological assays to know how your compounds work with the ion channels. And next one, I would like to mention about the cardiovascular safety, or we so-called the CPAP. So I think already we remember this figure. So considering about the instance and impacts. So the most important one is the QT prolong for cardiovascular safety. So this is a very high instance and high impacts target, and we really need to pay attention for the arrhythmic risk. So the main key point is about the hERG channel. So hERG channel is a potassium channel. So if the drug interfere or bind to hERG channel, on the cellular level, they could find especially on the cardiomyocyte, they could find a prolong of the action potential. And from the ECG, we can find the prolong of QT wave. So what's happened when QT wave prolong? So we can see clinically, we can see the so-called torsade de pointe, meaning the very severe arrhythmia, and can generate a ventricular fibrillation and cause cardiac arrest and sometimes death. So this is very risky and very dangerous. So all the compounds will follow the guidance of FDA S7B to test if the compound really bind to hERG channel. So we provide two different binding site for hERG channel to evaluate if our compound really bind to hERG, and if the compound binds to hERG, we really need to take consideration about the further cardiovascular risk. And talking about the cardiovascular risk, so sometimes clients, they want to know more about the potential risk, because not only the potassium channel, but also sodium channel and calcium channel, they can alter the heart rate or the shape of the action potential of cardiomyocyte. So some clients, they want to be very careful, so they also pick up sodium channel binding assay and calcium channel binding assays to make sure they have no effect on sodium and calcium channel as well. And similarly, if clients, they really want to know how the compounds work with ion channel, we can pass the drug to our US site to perform more dedicated electrophysiological tests to understand how the compound really work with the ion channel, and if there's a really risk of how we can avoid. And the next one, we would like to go through the cardiogenesis safety. So already we know for cardiogenesis study, so this is a very time-consuming and also animal-consuming test. So from the current suggestion, we need to take around 2-year study to perform like 50 to 70 animals per sex with 3 different dose level, like high, medium, and low, and also double control. So for the whole study, several hundreds or even thousands of animals should be used for this cardiogenesis testing. And also a lot of histopathological follow-up analysis is necessary as a golden standard. So this is very time-consuming and very costly assays. So recently, there are some voice in recent years, some voice thinking about, okay, how we can reduce the use of animal and how we can make some alternative tests by in vitro methods to accelerate this process, and also to find out maybe more potential cardiogenesis risk, especially on the 4 classes like genotoxicity, hormone dysregulation, immunosuppression, and chronic toxicity. So a lot of voice already raised, and people are looking for some alternative in vitro way to perform this cardiogenesis safety. Of course, so far we don't have a solid conclusion, so animal model is still necessary, but little by little, I think people will shift and think about more how we can address this issue by in vitro assays. So here we just summarize, check the literature, and we find some key characteristics of carcinogen. So there are some potential pathways like metabolic activation, genotoxicity and DNA repairment, and also epigenetics, oxidative stress, chronic inflammation, immunosuppression, receptor-mediated effects, and in the end, some cell immortalization and proliferation. So according to the reference, we pick up some assay lists related to different areas, different pathways. So like metabolic activation, ROS. So we have some free radical scavenger test, and we have an inflammatory related pathway, also apoptosis related pathway, and some important GPCR and NHR, and some GPCR related to cell proliferation. So by these molecular targets assays, so it will be helpful to our client to identify or to adjust the potential cardiogenesis risk. And in the end, I just like to show you the in type-based side, because type-based side is very unique. As a CRO, we have both in vivo and in vitro capabilities. So some clients, they start from in vitro studies, and later on they go through the whole process and go to the in vivo test for the animal study. So you can complete all the study in only one site. You don't need to ship your compound, and you have the same technical team to help you. Okay, so a very often phenomenon or question we met with our clients is about the safety pharmacology in vivo. And at the beginning, some clients, they finalize their in vitro study. They want to go to next step, some in vivo study. So they don't know the dose they want to apply to the animals. So this is the first study we suggest clients to do, is the maximum tolerated dose, so-called MTD. Because sometimes clients, they just pick up as dose, but when we apply to the animal, we find animal feel so uncomfortable, unwell, even in the end, lead to a death. So that's will terminate the study. So the first step, we encourage clients to do the maximum tolerance test to know how much dose we need to really apply to the animal, and then we can go to the next one, like some functional assay. For example, we have some locomotor activities, rotarod test, and also we have a cardiovascular monitoring like QC prolongation in guinea pig. And also we can monitor respiratory functions, such like respiration rate, tidal volume, mean volume, and also BRIC acids. And also in Eurofins type-A side, we also provide non-GLP based safety pharmacology and toxicology. So we have some toxicology studies to evaluate the vital clinical signs, to evaluate some adverse effects. So we can take the samples from blood, urine, or some tissue samples. And also we have some custom assay for nephrotoxicity and GI side effects. So in the end of this presentation, I just like to remind you in Eurofins, especially in our side, we can provide very distinctive value to our clients. In type-A side, we have more than 450 biochemical assays, so cover hundreds of potential molecular targets. And also we have more than 220 functional assays. And we have different technologies like radiometry, absorbance fluorescence TR rate, and also we have a confocal-based high-content imaging system, flow cytometry, Luminex, qPCR, and we have a very, very delicate cell culture system. And in recent years, also we introduced the SPR service to further study the binding affinity and kinetics for clients' compound. And not only for safety pharmacology, we have also therapeutic-based services. Like we have a therapeutic panel, like a NASH panel, T2TN studies, and neurodegeneration panels for on-target profiling assessment. So if clients, they are looking for a certain therapeutic area, we can also provide relative services. And in the end, not only for small molecule, we have also large molecule services. So for the new biotherapeutics or biologic study, so we have FC receptor binding assays, we have a C1q binding, and also we have some immuno-oncology targets like PD-1 and PD-L1. So this is newly launched in the past year. So this is the capability from type-A side. So just like to remind you, not only the safety pharmacology, but we have a lot of on-target profiling. So our staff, we have like more than 10 years experience, and more than 70% of our staff, they have a scientific background, and we have a very strict anti-deliver rates. And also we can support different kind of documentation or regulatory support to our clients. Yeah, so thank you for your attention. So we are looking forward to hearing from you in the coming future.